Hello everyone. Our project was titled Transcriptomic Alterations in Non-Small Cell Lung Cancer, which dealt with the impact of betel quid, alcohol, smoking, and tobacco exposure. My name is Jennifer Lawrence, and I pursued my master's in bioinformatics at St. Aloysius Institute of Management and Information Technology, Mangalore. My major focus in this project was to understand the impact of betel quid on non-small cell lung cancer.
Hello everyone, I am Pratiksha. I have completed my MSc in Bioinformatics from St. Aloysius College, Mangalore. In this project, I studied how alcohol exposure contributes to the development of non-small cell lung cancer.
Hello everybody, I'm Kritima. I completed my master's in bioinformatics at St. Aloysius Institute of Management and Information Technology, Mangalore. In this project, I worked on smoking impact in non-small cell lung cancer.
Hello everyone, I'm Sharon Xavier. I've pursued my master's in bioinformatics at St. Aloysius Institute of Management and Information Technology, Mangalore. My major focus lies on impact of tobacco exposure on non-small cell lung cancer.
Before we dive into the project, let's understand a few details about non-small cell lung cancer. It accounts for about 85% of all lung cancer cases, and it has three main subtypes: adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. Major causes of non-small cell lung cancer include smoking, exposure to pollutants, asbestos, radon, and diesel exhaust. Genetic mutations, alcohol, betel quid, and tobacco chewing also emerges as major cause. Moving on with methodology, our first step was data collection from GEO database. Our dataset ID was GSE30118. It was a microarray data and it had a total of 7 samples, among which 2 were normal samples, 2 betel quid samples, 2 alcohol samples, 4 smoking samples, and 3 samples of tobacco. Next step was dimensionality reduction process, which is also called as principal component analysis. It focused on log2 transformation and normalization. Next step was differential gene expression analysis using GEO2R tool, wherein we focused on identifying the potential upregulated and downregulated genes. Next step was enrichment and pathway analysis. Our major focus was KIT pathway and gene ontology analysis, which majorly included biological process, molecular functions, and cellular components. Next step was gene network analysis, wherein we analyzed for potential network relations in upregulated and downregulated genes in order to identify the hub genes in each of them. Next step was correlation analysis using GAPR2 tool, wherein we looked up for any positive correlation or negative correlation between the genes.
Coming to the result part, principal component analysis was performed to reduce the dimensionality of the complex datasets. Here we can see the clear separation between the betel quid exposed samples and the control samples, which indicates the difference in their gene expression profiles. In case of alcohol exposure, the PCA plot shows strong clustering pattern where the exposed and control samples are completely separated. We again notice a clear distinction between the smoking exposed and control samples with no overlapping. And finally, for tobacco, we observe similar trend where the exposed samples cluster tightly away from the control samples. This confirms that betel quid alcohol smoking and tobacco leads to strong and consistent transcriptomic differences. Next step was the differential gene expression analysis, which is done using GO2R tool. Here, in case of betel quid exposure, 630 genes were identified as the differentially expressed genes. Here, the downregulated genes are present on the left side of the volcano plot and upregulated are present on the right side of the plot. These are the genes which are highly regulated in case of betel quid exposure. Moving to the next case, alcohol exposure, where 259 genes were identified as the differentially expressed ones. Here, 135 genes are upregulated and 107 genes were downregulated. In case of smoking, 450 genes are identified as the differentially expressed ones. Genes such as PDP and PRDM14 are upregulated, and CA10 and RHAG were highly downregulated. Moving to the next case, tobacco, where 779 genes were differentially expressed. Among these 779 genes, 446 genes are highly upregulated and 312 genes are downregulated.
Coming to the pathway analysis using enrich-R, in betel quid exposure, we can see the significant pathways where the processing of the DNA double-strand break ends, HTR through homologous recombination or the single-strand annealing. Coming to the next sample, that is the alcohol exposure, it alters key pathways like O-glycan biosynthesis and hormone signaling, disrupting cell communication and promoting metastasis. Coming to smoking exposure, it activates DNA repair pathways like nucleotide and base excision repair due to tobacco-induced DNA damage. It also triggers immune-related pathways. In tobacco exposure, it is associated with immune dysregulation highlighted by enrichment in autoimmune thyroid disease and T cell signaling pathways. Coming to the gene ontology analysis, let's see about the biological processes. In betel quid exposure, it impacts both tumor suppressive and developmental pathways such as p53 signaling and bone morphogenesis. In alcohol exposure, it disrupts key mitotic processes leading to chromosomal instability and abnormal cell division. In smoking, accelerates non-small cell lung cancer by progression by promoting vascular remodeling, oxidative stress, and mitochondrial dysfunction. In tobacco exposure, it triggers strong cellular stress response, particularly the unfolded protein response, along with disruptions in ion metabolism and mitotic checkpoint signaling. Coming to the molecular functions, in betel quid exposure, it affects key molecular functions by altering epigenetic regulation and protein degradation processes. In alcohol exposure, it enhances molecular functions related to detoxification and lipid metabolism, such as aryl sulfotransferase and CoA carboxylase activity. In smoking exposure, it alters transcriptional regulation in non-small cell lung cancer by enriching molecular functions related to DNA binding, including sequence-specific and double-stranded DNA binding. In tobacco exposure, it significantly disrupts calcium signaling through enriched calcium ion transporter activity alongside altered protein modification and cell communication via D-sumo-oxylase and neuropeptide receptor binding. In cellular components, betel quid exposure affects both nuclear and membrane-associated cellular components, including chromatin remodeling complexes and focal adhesions. In alcohol exposure, it leads to enrichment in cellular component related to mitosis and ciliary functions such as condensed chromosomes and inner dynein arms, indicating disrupted cell division. In smoking exposure, it alters key cellular components including clathrin-coated cell vesicles and late endosome lumen, indicating changes in endocytosis, signaling, and immune invasion. In tobacco exposure, it leads to significant enrichment of clathrin-coated vesicles, indicating altered intracellular trafficking. Disruptions in nuclear membrane, lysosomes, and focal adhesion components suggest impaired nuclear transport.
Coming to the gene network analysis, in this we'll be seeing betel quid versus normal, as well as the gene network between the upregulated gene network as well as the downregulated gene network. In case of betel quid, we have PARP11. It emerges as a central hub gene, and it has a close interaction with PARP family, as well as there is under close connection with other genes. In downregulated gene network, we have gene as the TSPYL4 as a hub gene, and also close association with multiple TSP5 family members. In case of alcohol, In upregulated gene network, we have PAX2 as a central hub gene, and it also has interacting genes such as WTI and others. In downregulated gene network, we have WDR33 as a hub gene as well as the other genes. In case of smoking, we have PDPN, that is podophyllin, as a central hub gene. In downregulated, we have EML1 as the hub gene. In case of tobacco, in upregulated, we have HMCN1, that is hemicinthin, as a hub gene. And this hemicinthin is nothing but it's a structural protein that helps hold tissue together and keep cells properly organized, especially in organs like tissue. But in case of NLCL patients, it behaves differently than it should be. Coming to the downregulator, here we have RAI14, that is retinoic acid induced 14. This acts as a hub gene. Coming to the correlation analysis, this is nothing but a statistical view of how the two genes interact within each other. And we have positive correlation and negative. Positive, whereas the two genes are co-expressed together, where both are increased together as well as decrease. Whereas negative, the one increases and the other decreases, and the vice versa. In case of Betel Quid upregulated correlation network, the correlation between the PARP11 and NAV3, here we have a positive correlation. In downregulated, we have TSPY L4 and PDNP, we have a negative correlation between these two genes. In case of alcohol, as well as upregulated and downregulated, there is no correlation analysis between this. In smoking, upregulated, we have a correlation, a weak positive correlation between the genes such as PDPN and TMDI-M4. In downregulated, we also have a weak positive correlation between the genes such as EMLI, L1 and PACSIN2. In tobacco, in upregulated, we have a negative correlation between the hemicinthin and YME1L1. We have a negative correlation. That means the two genes are not co-expressed together, where one increases and the other decreases. In downregulated, we have a weak positive relationship between the retinoic acid induced 14 gene as well as PACSIN2 gene.
Apart from all these analyses, we also conducted comparative analysis in order to find out common genes and common pathways. Comparative analysis was done in order to identify common differentially expressed genes. Tobacco had a unique gene as of 451 genes, betel quid 116 genes, alcohol 141 genes, and smoking 38 genes. A total common of 10 differentially expressed genes were identified. These are the potential roles of these 10 common genes in non-small cell lung cancer. Comparative analysis was conducted to identify common pathways. Wherein tobacco had 44 unique pathways, betel quid had 10 pathways, alcohol had 21 pathways, and smoking had 2 pathways. Wherein altogether we had 10 common pathways in each of the from among all the 4 conditions. These are the 10 common pathways in all the 4 conditions, that is betel quid, alcohol, smoking, and tobacco exposure. Concluding our work was basically in order to understand and explore the impact of betel quid, alcohol, smoking, and tobacco exposures on non-small cell lung cancer development. Principal component analysis revealed that there are no clustering and there is clear separation between exposed and control samples. A pathway and enrichment analysis showed the enriched pathways in among all the 4 conditions. Network analysis was conducted in order to find out any potential network between all of these genes as far as upregulated and downregulated genes, and also we found out the hub genes in each of the conditions. And then we conducted correlation analysis in order to find out which of the genes were potentially positive correlation and negative correlation. Thank you.